Hey guys, welcome back to Blackboard Biology. In the previous video, we went over the kind of overview of cellular respiration, and in this video, we're going to dive down into the nitty-gritty with aerobic respiration. Again, this is the type that uses oxygen. So I kind of left you here in the last video that what we're going to do is we're going to see that cellular respiration, the products of it are going to come out and they're going to become the reactants of photosynthesis, and what comes out of photosynthesis will go into cellular respiration. Specifically, we're going to have CO2 and water come out of respiration going into photosynthesis, and out of photosynthesis. Now, again, remember, it's not really glucose. It's actually G3P. It's a smaller sugar that can be, two of them can be combined into glucose, but that's what we say it comes out just to make it nice and pretty. And oxygen gas, which are going to be going into cellular respiration if it is aerobic. And so the whole point of this process is to take glucose over here on the left, which has tons of energy packed into its chemical bonds, and we want to take some of its electrons away, and we want to use some of the energy that's trapped up in those chemical bonds to produce this molecule over here, ATP, adenosine triphosphate, because what we can do with it is we can break its bonds here, break off this phosphate, and we can release lots of energy by doing so. And so what we want to do in the process of cellular respiration is to take glucose, put it through the process, and produce lots and lots of ATP. With aerobic respiration, we can do that. So as I mentioned in the previous video, it's a quick recap here, cellular respiration can occur two different ways. Aerobic with oxygen, that's the point of this video, or anaerobic with no oxygen, and I'll do a separate video for that. So this video, we're going to dive into specifically aerobic respiration, which has 3 steps, and we'll go through each step in detail. But before we get there, we need to kind of discuss, where is all this going to occur in the cell? Well, here we go. The mitochondrion, okay, also often referred to as the powerhouse of the cell. Now, if you know nothing else about organelles, you probably know that. In fact, I like to show my students memes and things about the mitochondria. Mitochondria is plural for mitochondrion. And they often say, things like, I see memes that say things like, students asking questions to the school system, what are taxes and how do we pay them? And the school system says, fear not, the mitochondria is the powerhouse of the cell. And it's as if that's the one thing you take away from high school sometimes, is that the mitochondria are the powerhouses of the cell. You may not know anything else, but you'll remember that. So I hope that through this video and other things, you'll come to learn more than just it's the powerhouse of the cell. But let's kind of break it down a little bit. So the mitochondria are these wonderful little organelles that you have in your cells. You have a lot of them in your muscle cells, and they are actually double membraned. So here we're going to pretend that there is a membrane on the outside. I didn't really draw one, but we're going to pretend there's a membrane on the outside, and then this white is the inner membrane. It's going to be very important here in a moment. So that's the inner membrane, and then in here, this is known as the mitochondrial matrix. So yes, we will be entering the matrix in this video. And so this is very important. Now, again, this is within the cell. This organelle here is within the cell, sitting around in the cytoplasm. So again, this space out here on the outside, here's the outer membrane again. And all this space out here on the outside would be cytoplasm, or sometimes referred to as cytosol. Okay, and it's in the cell. So all this space around is the cytosol, and that's going to be important because here in a moment I'm going to tell you where each step of aerobic respiration occurs. And so you'll notice that this inner membrane of the mitochondrion is highly folded. Now, this is just my very, very simplistic representation of it. It's actually got way more folds than that in that inner membrane. And the purpose of that, all of those folds increase the surface area. And by increasing the surface area, we increase how much electron transport can occur, and we increase the amount of by increasing the amount of electron transport that can occur, we increase the amount of ATP that we can very rapidly produce in those mitochondria. So it's very, very important. And so now that we kind of have a little bit of understanding of the mitochondrion and kind of its shape and this inner membrane that's highly folded and the matrix, let's dive in a little bit into the steps of aerobic respiration, and then I'll show you each one individually. So I'm going to move quickly, so if you need to, feel free to pause the video, rewind, do whatever you need to do to take your notes. So aerobic respiration occurs in 3 steps. So let's start with glycolysis. So glycolysis is going to be the breakdown of glucose. Break apart glucose. Okay? And so what it does, when it breaks apart the glucose, I'm going to tell you how many ATP, how many adenosine triphosphate molecules we get from each step, because that's our whole purpose of doing this, is to make ATP. So how many do we get from glycolysis? Well, in total, we only get 2, which is not a lot. And so, but what we're doing here with glycolysis is we're taking glucose and we're putting it through this process, breaking it apart, and I'll tell you what it breaks apart into in just a moment. And we're also producing some ATP, and we're producing some, or I shouldn't say producing, we're giving some electrons to electron-carrying molecules. Now, if you watch the photosynthesis videos that we did, you'll recall that there are molecules that are carriers for electrons, and that is how we're going to get those electrons to the electron transport chain. So let me tell you, glycolysis, here's what it needs. Here's what we need to get glycolysis going. Of course, we need glucose, because I just said we're going to break apart glucose. We also need what is known as NAD+. It's one of those, the empty, quote unquote, empty form of an electron carrier. It doesn't have those extra electrons yet, so it's an empty electron carrier. And we also need some ADP, right, and some phosphate. And so those things are what we're going to put in and out. So we'll say produces. The glucose is being broken apart into 2, I started to misspell that, into 2 molecules of pyruvic acid. Okay, those are 3 carbon molecules. Glucose has 6 carbons, and so here in a moment, to simplify things down, I've shown a representation of these molecules just by their carbon atoms. They're more complex than that, but it makes it simpler for our purposes. And so pyruvic acid is a 3 carbon molecule, and so 1 glucose molecule will make 2 of those. We will also produce NADH. Okay, so when we give that pair of electrons to NAD+, it also grabs a hydrogen ion and it becomes NADH. That is the carrying version. It has 2 extra electrons, an electron pair that it will take to electron transport chain. And then we also produce some ATP, of course. Okay, so that is glycolysis in a nutshell, real fast. We'll go in depth here in a moment. And then after that, we'll go to the Krebs cycle. Now, the Krebs cycle is also sometimes known as the citric acid cycle. And it truly is a cycle. It actually regenerates some of its own starting materials that it needs. And so it's called the citric acid cycle because Krebs is the name of one of the scientists that discovered it and kind of worked with it and everything. And citric acid is one of the things that are produced during the cycle. So the Krebs cycle is also only going to, and perhaps I need to maybe insert a line here so we can keep these nice and neat. And I'll go ahead and give myself another one to put down here and kind of block myself off a little bit. And so it is also going to produce CO2 net ATP from the process. Okay, and what is going to occur in the citric acid cycle? It's very complex. We're not going to go too in depth in this video, but here are some of the things that it needs. Okay, what goes in? We're going to take that pyruvic acid from glycolysis. All right, so we've got pyruvic acid, and we are also going to produce, as I mentioned, some ATP. So we're going to need ADP plus that extra phosphate. We'll also make some more NADH, so we'll need NAD plus. And then there's actually a second electron carrying molecule. FAD is its empty form, and when we give it the 2 extra electrons, because you'll notice FAD does not have a plus with it. It's not positive. It's already a neutral atom, or excuse, not atom. It's already a neutral molecule, excuse me. It's already a neutral molecule. And so when we give it those 2 extra electrons, it actually picks up 2 protons so that all things remain balanced and neutral. And so it becomes FADH2. I'll write that here in a moment. So what we produce with the Krebs cycle Okay, some of what the Krebs cycle produces, of course, as I mentioned, we're going to make NADH, FADH2, and ATP, but this is where CO2 is given off. So part of the process of converting the pyruvic acid and breaking it apart, it gets converted first into citric acid, and even before that, actually, the pyruvic acid is technical term, it's oxidized, and what it does, it actually releases a CO2 molecule. And then again, when there's one of the things it's converted to is called acetyl-CoA. We'll get into that in a moment. That combines with a 4-carbon molecule to become citric acid, and then that is broken down twice, broken down into a 5-carbon molecule and then broken down again into a 4-carbon molecule, and each time that releases a CO2 molecule. And then, as I mentioned, we also produce ATP, NADH, and FADH2. That is the electron carrying version of FAD. So, if you'll notice kind of what we're doing here, we're building up some things to ultimately get to electron transport chain. Now, the electron transport chain, I'll go ahead and tell you, this is the one that needs oxygen. That is what makes all of this aerobic. Glycolysis can occur in an anaerobic fashion where the NADH produced in it will go and play a part in fermentation, and the pyruvic acid will play a part in fermentation. That's anaerobic. The Krebs cycle, what is produced out of it and the way that it functions, it must occur alongside of the electron transport chain. And so because the electron transport chain needs oxygen, we say the Krebs cycle is also aerobic, even though it's not where the oxygen comes into play. So the electron transport chain is going to need oxygen. It needs NADH because it's carrying those electrons. It needs FADH2 because that is also carrying electrons. And then something else that it technically also kind of needs is hydrogen ions, which there are plenty of. Because again, if you watched the photosynthesis video, the same way that the electron transport chain works in photosynthesis, we're going to pump those hydrogen ions against the concentration gradient and build up this this great potential difference. And because those hydrogen ions want to move back and spread out and get away from each other and go back the opposite direction, that's going to power some ATP production. Okay. And so what does the electron transport chain produce? Well, this is the moneymaker right here. So it produces, of course, ATP, It actually produces water. That oxygen gas gets combined with hydrogen and makes water. And then, of course, after we take the electrons away from NADH, we're left with NAD+. We're left with FAD when we take its electrons away. And so what it does, it produces a total of 32 ATP. So this is the moneymaker. This is where it occurs. This is where it's all at. So we really need to get to electron transport chain. That is where we get the most ATP. So in total, if you add these up, we have 2 in glycolysis, 2 in the Krebs cycle, and 32 ATP in the electron transport chain for a total of 36 from one glucose molecule. Okay, so one glucose molecule can net us 36 ATP. And those ATP molecules are ready to go. They'll provide lots of cellular energy. And so this is very, very important to your body, especially to your brain cells. Your brain cells need lots and lots of energy. You'd be surprised how much energy your brain actually uses to keep everything in your body running. And so that's very, very important. That's why you sustain brain damage if you go without oxygen for too long. Because those cells need lots and lots of ATP. When those processes that run on ATP stop happening and kind of break down in your brain cells, you sustain damage to them, which obviously is very, very bad. Your muscle cells also require a lot of energy when you are moving and exercising and doing things. And so your muscle cells would much rather do aerobic respiration with plenty of oxygen. If you don't do aerobic respiration, if you use up the oxygen in your muscle cells too quickly, they'll have to resort to using anaerobic respiration. And that's how you create that oxygen debt, so to speak, where you're breathing really heavy after sprinting or lifting weights or doing something of that nature. So this video is already getting a bit long, so let's dive right in and get into the specifics of each step, and I'll try to make this quick. So step 1, glycolysis. So glycolysis is going to occur outside of the mitochondria. And so let me write here. This is all occurring in the cytosol or cytoplasm. It's all out there. It's outside. So this is happening in the cytosol outside of the mitochondria. So again, this can occur without oxygen. It doesn't have to be an aerobic process. Glucose is going to be broken apart into 2 3-carbon molecules. That's what you see occurring right here. All right. And in that process, we actually have to use 2 ATP first. So to break apart the glucose, we actually have to spend money to make money, so to speak. We're going to use up 2 ATP first to get that split apart, but then the way that it gets converted, it will become 2 pyruvic acid molecules. And the process that goes through, we're releasing energy at that point. So we use some energy to break it apart at first, but then we're also releasing some energy, and we're also giving away 2 pairs of electrons over here. So these 2 NAD plus become 2 molecules of NADH. Now remember, these electron carriers are going to go to the electron transport chain. Okay? Now remember, and I don't think I told you this yet, but the electron transport chain actually occurs on the inner membrane of the mitochondria. Okay? So again, that's this over here. Right here's the inner membrane, this white, okay? That is where the electron transport chain will occur. So I really could, I suppose, kind of draw this. These are going over here, okay? They're going to the inner membrane. The pyruvic acid is going to go to the inner mitochondrial matrix. That's where it will happen. Again, I mentioned we're releasing energy, so 4 ADP become 4 ATP. So we spent 2 to get it started, but we gained 4, so we have a net gain of 4 excuse me, I'm trying to erase some things here. So we have a net gain of 2 ATP overall. So again, glycolysis occurs in the cytosol, and we gain 2 ATP. Okay? So this pyruvic acid is going to go into the inner matrix, and we'll be moving on to the Krebs cycle. So here's the Krebs cycle. It's a little bit more complex, but here we're going to start up here with our pyruvic acid from glycolysis. It's going to go through the processes where it gets converted, and it actually will be broken apart, and 2 of those carbons will become a molecule here. One of them will be combined with oxygen to make CO2. Okay? Now the Krebs cycle, as you can see here, is occurring in the inner matrix, the mitochondrial matrix. That's all this pink I've got colored here. Mitochondrial matrix is where all this is occurring. And so we were up here in this part, and we're moving acetyl-CoA. All right, is the molecule that is produced, and it will combine right here with a 4-carbon molecule that's already in the cycle. Okay, so the cycle already exists. The cycle is already there. And so there's a 4-carbon molecule that's going to combine with acetyl-CoA to produce citric acid. All right, and so that citric acid is then going to be broken down into a 5-carbon molecule and release some CO2. That will get broken down again into a 4-carbon molecule and release CO2. Apparently I didn't draw my other CO2 molecule that should have been released from that. And then that 4-carbon right here, that 4-carbon molecule will continue through this cycle, through these intermediate steps, until it eventually gets back to the original. It regenerates itself to the original 4-carbon molecule that we need to combine with acetyl-CoA. So it truly is a cycle. I apologize. I did not mean to erase all of that. I only want to erase some of the ink. A little bit struggling here. Here we go. Oh, I don't want to erase all of that. Okay, I'll just leave it. So, this is a cycle. And if you'll notice as we go through this cycle, there are going to be some things that we produce. So, again, electron-carrying molecules. We will produce NADH. We will produce FADH2 over here. FAD picks up a couple electrons, becomes FADH2. NAD plus picks up a couple electrons at different points along the way, becomes NADH. And we also produce some ATP. Okay, so it occurs in the mitochondrial matrix, and we're going to get a net gain of 2 ATP from this as well, per 1 glucose molecule. However, you'll notice this all starts with just 1 pyruvic acid up here at the top, but remember we got 2 of those from glycolysis, so we'll get 2 turns of this cycle. All right, so rolling on. These are the big things that we got from here. 2 ATP, it's not bad, it's good, but the NADH and the FADH2 are really what we want because we want to get to electron transport. Okay, so what we are looking at now is over here is our mitochondrion. So we're looking at this little cutout, blown up section, and I've drawn these electron carrier proteins. Okay, that's what these purple things are. I don't think I wrote that in there. These are electron carrier proteins. All right, all the purple things there are electron carrier proteins. And so what we needed to do was produce NADH, FADH2, so that they could carry those extra electrons to the electron carrier proteins. Now, these are high energy electrons. We're giving it to them, and they're going to move through the electron carrier proteins and gradually make their way to lower energy states, giving off energy as they do this. So that energy, and this is the remarkable thing about the electron transport chain, that energy will be used to pump hydrogen ions, all right, our little blue dots here, hydrogen ions into this intermembrane space. So if you'll recall, earlier I said this white right here, that's the inner membrane. That's where all these proteins are embedded. And then out here we have the outer membrane. Well, all this red space, the intermembrane space, we've pumped a lot. You can see a lot of these, right? These ions are pumped in here. That's a good thing. That's what we wanted to do because they naturally want to get back across this membrane. They can't. There's no pathway for them to do that except really through this ATP synthase protein over here. So what they do, they will come and they will go through this protein back in here where there's less of them. That's where they want to go. When they do that, they're energized enough. They move through there with enough force because of the concentration gradient. They move with enough force that they make this ATP synthase protein spin, and that actual movement of it is enough to combine a phosphate back with this ADP and produce ATP. And if you'll recall, remember I said we are going to be able to do that enough to produce 32 ATP from this process. So what happens to those electrons as they're given to the electron transport chain? Well, if you'll follow these yellow arrows here, right, they go through, these electrons go through, and they come back, and they are actually combined, those electrons are given back to this oxygen and these hydrogen ions here, which is, again, a hydrogen ion is just a bare proton with no electron. And so what those will do, the 4 hydrogens will combine with these 2 oxygens from an oxygen molecule and become water. Well, remember, this whole video is about aerobic respiration, which requires oxygen. This is where that comes into play. It requires oxygen gas. This is why you must breathe. This right here, this is the very reason that it doesn't feel good when you hold your breath for too long, because your cells are trying to do this. And so that's very crucial to keep the whole cycle, to keep everything running, and to produce lots and lots of ATP. You need that oxygen for this process to continue to occur. Okay, so please continue breathing, take in oxygen. And so if I could, just to very quickly, let's go back and kind of recap the overall steps. Glycolysis, then Krebs cycle, then electron transport. In glycolysis, we're breaking apart glucose. We get 2 ATP. The Krebs cycle, we're going to produce citric acid. All right, as part of that, we're going to put it through all this cycle. We're going to produce 2 ATP, and many of our electron carriers give off CO2. Finally, we get to the electron transport chain, which is where we really want to get to, because we get 32 ATP from that, and then it basically takes all of these things and puts them back into the cycle overall. And so I know this was a long video, but I hope that it was very helpful to you, and I encourage you, comment if you have questions, subscribe to my channel, and if you're any of my Scott High students, feel free to reach out to me at any time. Thanks for watching.